Among his work, he's probably best known for his attempts to illustrate some of the masterpieces of historical literature. We see here his rendering from Milton's Paradise Lost called The Fall of Man. It depicts Milton's description of humanity falling into hell, what Milton called darkness visible. I love that turn of the phrase. It's doing what good art should do. It's trying to communicate something that we don't quite have the right language for, right? That we don't have the right words for. Another person who liked that turn of phrase was a guy by the name of William Styron. He's an author, he's a novelist. If you know him, you probably know him from his novel called Sophie's Choice. Right? What you may not know is that after writing Sophie's Choice, many years later, he fell into a deep depressive episode. Hospitalized, nearly took his own life, emerged out of it, and several years later wrote a book called Darkness Visible, where he described in some of the most engaging and lucid detail the actual experience of being depressed and coming out of that depressive episode. I'm sorry to insult you by reading passages on the screen here, but I don't know how it's showing up online, so I will. In depression, this faith in deliverance is absence. The pain is unrelenting, and what makes the condition intolerable is the foreknowledge that no remedy will come, not in a day, an hour, a month, or a minute. It's hopelessness, even more than pain, that crushes the soul. This really moved the needle in the national conversation about mental health when it came out. I want you to hear in their own words from some patients that are experiencing a major depressive episode.
It was kind of like sliding down into the bottom of a well. Very, very dark. There's no one there. There's really no light.
Feeling, I look in the mirror, I don't see the person that I know as me. It's like a stickiness. It kind of holds me down, and it takes so much effort to do anything.
Very heavy. The worse it gets, the heavier it feels on me. Like I can't move. Sort of.
Sorry, I know the audio is probably quiet here in the room, but hopefully you could hear much of that. There are a few things that I hope you can take away from that. One is the way that they use analogy to describe what it is that they're feeling. They don't quite have the right words for it, but they keep using it's like this, it's like that. Also, the physicalness of the analogies. I mean, they talk about it in physical terms. Can't move, bottom of a well, things like that. Also, I just thought you can see it on their face. Here's one of those patients, a clip from that video, and a clip from that person a year later when they're out of that depressive episode. You can see their face during the depressive episode almost looks Parkinsonian, right? It's a flat affect, no emotion. You can see this in Styron's writing, too. Instead of pleasure, I was feeling in my mind a sensation close to but indescribably different from actual pain. The pain were readily describable. Most of the sufferers from this ancient affliction would have been able to confidently depict for their friends, loved ones, physicians, some of the actual dimensions of their torment, perhaps elicit a comprehension that's been lacking. For myself, the pain is most closely connected to a drowning or suffocation, but even these images are off the mark. I fell onto the bed and lay gazing at the ceiling, nearly immobilized and in a trance of supreme discomfort. So you see the same things. You see him saying, I don't have the right language for this, but I'm going to try through my analogies. You see him describing it very physically. Inability to move, drowning, suffocation. It's a disease of the brain and the body. And finally, unfortunately for me, you see that he's actually telling us some 30 years ago that dimensionality reduction is going to be the answer here. He's asking us to find some of the actual dimensions of his torment. So Fortunately, he's giving us some algorithmic guidance too. What I'm going to describe to you today is an effort that, in the context of depression, but I think applies more broadly, an effort to try and peek inside the black boxes of these complex systems. So if we take a step back from depression for a second, I would offer to you that I think a lot, maybe not all, but a lot of neuroscience experiments have a similar sort of set of challenges to them. We start with some external factor. If you're a sensory neuroscientist, this is a stimulus. If you're a motor neuroscientist, this is some task. If you're an affective neuroscientist, this is some environmental factor that's causing a change in you. But it's measurable. It's external. It's your stimulus parameters. You have it. This goes inside a black box of biological intelligence that has multiple components to it. As neuroscientists, we often think of the second of those, the neural activity, and how that's a black box that we want to peek into. And that's a technological challenge of being able to record from neural populations that we've made great progress on as a community. I'm going to focus more on some of the algorithmic challenges. Often what we do is we try to correlate or correspond our neural activity to those external factors that we have nice and quantified. More and more we're doing that with advanced algorithms, what we'll call kind of broadly artificial intelligence, right? These are very, very powerful algorithms, right? But they're a little inscrutable in many cases. We don't know why they're behaving, how they're behaving, which for commercial application has all sorts of issues of trust and fairness and equity. But in a scientific domain, it limits our scientific understanding that can come from these algorithms sometimes. We would really like to be able to explain what's going on in those algorithms. And in fact, that's a big focus of the next generation of artificial intelligence tools, called the so-called third wave of artificial intelligence tools, where we look toward things like explainability, right? We try to add on to these tools or develop them from scratch to try and give some information, some explainability about them. I'm actually going to offer that there's another black box that we hope to peek inside in this diagram that we don't often think about much, and that's the internal thought, I'll say very broadly. Again, if you're a sensory neuroscientist, that's the internal perception. If you're a motor neuroscientist, that might be the task motivation, the task intention, I should say maybe. We often don't try to correlate our neural activity to this because, frankly, we often don't know it. We can run behavioral experiments sometimes, but those are very limited, right? And they're limited for a few reasons. One is that we don't necessarily know what the natural feature space is for these internal thoughts. So when we're working with animals who don't have any language, it's kind of very difficult to know exactly. So we use very impoverished behavioral measures that often correlate to our measurement variables of the external factors, as best as we can. Even with humans that have language, though, we just saw in cases like this, where we're talking about interoceptive feelings, affective feelings, they don't always have clear language for it. So we're just going to touch on that briefly today. We're going to spend most of our time talking about explainable artificial intelligence. But at the end, I'm just going to tell you a little bit about some new tools that I think are actually going to help us peek inside this other black box a little bit as well. We'll spend most of our time here today trying to peek inside this black box and use some tools from this next generation of artificial intelligence tools, explainable AI tools, in order to help us better understand recovery from depression under deep brain stimulation. I want to make sure to cover the most important slide first. It is a joy to work with all the collaborators that I get to work with. I have many of them listed here. I want to call out a few specifically. One is Shankar Agapan. He's a research engineer that I work with here, also works with Rob Butera, and many of the things that I'll show you today are due to his skilled hands. Of course, the patients and the clinical team that cares for them. Some key collaborators from Mount Sinai, Helen Mayberg, and two people on her team, Ki-Sung Choi and Steve Heising, which did some of the key analysis that I'll show you as well. And Patricio Riva-Pose, who is the co-lead of this team, along with Helen and I, on the project that I'm going to tell you about. I want to make sure to tell you about any potential conflicts of interest that could color what I'm going to tell you today. I won't belabor this because I think everyone probably has a good appreciation for the cost of depression, but just to put some numbers to it, for non-communicable diseases, neuropsychiatric disorders are almost 30% of the overall disease burden, about 10% of that comes from major depressive disorder, clinical depression, also called unipolar affective disorder. It's an enormous disease burden. Okay? Today, there are probably 300 million people globally that suffer from major depression. In the US, it's estimated that the cost just to employers is over $100 billion per year. It's a leading cause of disability, certainly in the developed world. And there are 20 to 30% of these patients that are treatment resistant, meaning that at this point, there is no approved therapy that gives them relief from their symptoms. Okay? We're in the middle of a presentation right now, so we'll handle it another time. Thanks. I think no one needs convincing that this is an issue to work on. And like my family, many of you have probably been touched by depression in your own families. I don't want to belabor this, but again, just to tell you that for me personally, there are two communities of people that I care about deeply that are affected by this. One is that there is a huge influence of income level on diagnosis of depression. Huge influence, right? Again, not to put too fine a point on it, but I grew up here. I grew up in poverty, and much of my family remains in poverty. This is my grandmother. She's 96 years old. That's a picture from last summer. I point her out because she is the reason that I grew up in a family of Native American heritage. She's an active member of a Native sovereign nation a few miles from where I grew up. Native American mental health has a 2.5 times suicide rate for their young adults. Their children and teens have the highest major depressive disorder rates of any ethnic or racial group in the country. 22% of Native American females have attempted suicide. Almost 1 in 4 people. Astounding number. So again, I don't think that I need to probably convince you that it's an important problem to work on. Let's talk about what it is and how we diagnose it and how we treat it. These will be important things as we try to examine it. This is the DSM-5 diagnostic criteria. This is how depression is diagnosed. There are 2 core symptoms, depressed mood and anhedonia, a lack of an ability to experience pleasure or joy. You must have one of those 2 core symptoms over an extended period of time. You must also have some of the additional symptoms, weight loss, decrease in appetite, insomnia, psychomotor retardation is the common one, psychomotor slowness, fatigue, loss of energy, guilt, worthlessness, diminished cognitive capacity, ability to concentrate, thoughts of death, suicidal ideation. So clinically, it's diagnosed by matching these criteria through clinical interviews. There are ways that we try to measure the severity of depression. I'm going to give you an example of one scale here. It's called the Hamilton Depression Rating Scale. It's probably the most common of these. These are subjective survey instruments. So a clinician would fill out a survey after meeting with you and answer, in this case, 17 questions about how you're doing. I want to be very clear. This is not a diagnostic for depression. It was never intended to be. This was developed in the 1950s. It was developed to measure how well inpatient depression patients were doing in response to the first generation of antidepressant drugs. Okay, so it's very focused, as you see here, on the physical symptoms, right? If you look at the core depression symptoms, depressed mood and work and activities, which is essentially, are you doing things that you find pleasurable? They're 2 of the 17. There's a lot of stuff on here about different types of anxiety. Insomnia has 3 categories on here. So what you can see is this is a measure that's heavily weighted by a lot of different non-specific symptoms, many of which occur in the comorbidities that often come up with depression. So this is a numerical rating score. It is often used in drug trials, but it is not meant to be a diagnostic, and it's not meant to be applied all the time. Originally, the guidance was every 2 weeks you do this. Right now, the way to use this is at the beginning and end of, say, a drug trial to measure whether you've made an improvement overall. Okay? But it can be a deeply confusing measure with a lot of subjectivity. Major depressive disorder is a network disorder. We aren't necessarily going to go through this diagram in a lot of detail, but just wanted to show a few references that state kind of very clearly some of the major symptoms of depression have entire networks associated with them. So anhedonia, the loss of pleasure, deeply impacted by the reward network. So areas like nucleus accumbens, things that modulate dopamine production in the brain. Cognitive control has both limbic and default mode network elements impacted. Dysphoria, so feelings of unsettledness, like something doesn't feel right in my body, has a lot of impact from the limbic networks as well. Rumination, default mode network, comes up a lot. It's kind of internally focused thought. You see here on the right that these networks, these major kind of networks, are modulated in depression. Default mode network seems to increase its activity, so more inward focused thought. Things like the salience network that helps you assess sensory motor information and adjust cognitive control seems to be downregulated. Dopamine is downregulated. The serotonin and dopamine networks are deeply connected to all of these areas that seem to be mediating the symptoms that we're showing you here on the left. Right? These are often found through correlational studies. I think the picture that I want you to take away is it's a network disorder and it's complicated. You know, it's difficult to know how these pieces all fit together to get a picture of what's going on. While there are many depression treatments, I'm not going to go through the history of that. We're going to focus today on deep brain stimulation in the subcallosal cingulate. So this is an experimental therapy. It is meant for treatment-resistant patients. It's obviously very invasive. This was pioneered by Helen Mayberg. The first paper reporting this was in 2005 with a cohort of 6 patients up in Toronto. Again, there's a really, really interesting long history to this that I won't be able to go through today. But I'll just say that they identified a target called the subcallosal cingulate or the subgenual cingulate. They did it in a data-driven way from imaging data, PET imaging mostly at the time. And they implanted deep brain stimulators and have done so in several cohorts of patients as they refine the therapy up to the present day. The current target has been defined also in a data-driven way. It's done now through individualized DTI imaging on the patients, but was found through a common map of responders in large cohorts. The key thing to take away are there are 4 white matter bundles, and what we're actually trying to hit is the intersection of those 4 bundles. There's in yellow there the cingulum bundle, in red the forceps minor, in blue the uncinate fasciculus, and in dark blue there, it's hard to see in this view, but frontal striatal fibers that hit subcortical structures. Just to help a little bit, we drew a quick diagram of this. So you can see in the frontal striatal, you're hitting subcortical things, thalamus, nucleus accumbens, very important in arousal and reward regulation. Aqueductal gray, autonomic function, raphe nuclei, critical nucleus in the serotonin network, seems to be involved in homeostasis of serotonin. In the limbic network from the uncinate, you have things like insula, hippocampus, amygdala. In the cingulum bundle, you have anterior, mid, and posterior. There seems to be some disagreement about how many divisions there are in the cingulum bundle, but certainly affected in things like cognitive control, pain, movement to some degree. Right? And then the forceps minor headed out to prefrontal cortex, especially ventromedial, so headed to the frontal pole. So you see key areas in both the salience network and the default mode network. So it's not clear that subcallosal cingulate, SCC, is actually the site of dysfunction, but it is a network way station, essentially, where a lot of really important nodes are crossing through. So it's unclear exactly where the deficit is, but this is the target that was identified, and people are getting better from it. What we see, and this is a longitudinal study over a cohort of 28 patients from Emory, where the majority of patients get better and stay better. You can see in this cohort they have over 75% long-term are clinical responders. The response in this case is defined by a 50% reduction in their symptoms according to a scale like the Hamilton. And they have about 50% of people that are in complete remission at the 8-year mark. This stands in contrast to the fact that actually this was the subject of a clinical trial earlier that was halted midway through. I want to be sure to use the right word there. It was halted with an indeterminate outcome. The breathless headline said the trial failed. They actually never got through to collecting their data. The company that was running it decided partway through, they ran a futility analysis and they halted it. There are probably many reasons for that. Again, if you want to talk offline, I can talk about them. There are a lot of sources of variability, and some things have been done since then to try and reduce that variability. A key thing is that they needed a more objective way to assess what was happening. If you look at the longitudinal data, they had a number of patients, I believe it was almost 75 patients, that kept the implant in. They turned on the controls, they turned on the sham group, so everyone was receiving stimulation. 2 years into stimulation, they actually had a 50% response rate. This is after the trial was halted. So response seemed to take longer, may have been due to imprecise targeting. Okay, that's a story for another day. The key thing is they didn't know that they were on their way to a good result. If you're using the Hamilton as an indeterminate measure, there are a lot of things going on. They needed an objective way to know that these patients were headed in the right direction, or if they weren't, to be able to do something about it, like adjust the dosage of their device. What we're searching for today is a potential biomarker here, an objective way to measure how a patient is doing through this therapy that would enable closed loop modulation. I know some of you work on closed loop modulation. This might be particularly slow closed loop modulation, like adjusting dosage on week timescales. Could also tell you when to administer adjunctive therapies, like new medications, psychotherapy, both of which are important parts of getting people better in deep brain stimulation. What we need here, we need two critical things out of a putative biomarker. We need it to correspond to a relevant behavior that we're trying to measure, and we need it to move when we apply therapy effectively. Those are kind of our two baseline things. If we're going to have a biomarker, it's got to measure something about what we want to capture, and it's got to move when we apply a therapy that is effective. So we'll keep those in mind. The primary data I'm going to tell you about is a cohort. We implanted 10 patients. These are treatment-resistant depression patients. They're implanted at Emory, implanted with Medtronic PC Plus S device, which is it was an experimental device. It was never commercially available, but it was the first device that really let us record from the stimulation site.
Oh, this was the one. Okay.
During therapy. So over 6 months of therapy with patients. We ended up with 6 patients of usable data. So everything I'm going to report to you from here on out today is an N of 6. So a reasonably sized cohort for an invasive procedure like this. They were all done with the kind of standard procedures, the state of the art in how this team knows how to do it. So individualized DTI targeting. You can see here on the upper right, this is a rendering of one of the patients. We can measure what the volume of tissue activated is from stimulation. We can measure the volume of tissue recorded. It's a differential recording on 2 electrodes around the stimulation contact. We can do computational modeling to understand what fiber bundles are being engaged, especially in the stimulation. These patients came in weekly for their clinical interviews. During the time that they were in, we collected data from them, 7-minute long recordings. We did stimulation on and stimulation off. Everything I'm going to show you today is with the stimulation off, so there is no possibility of a stimulation artifact in what I'm going to show you. Broke that up into 10-second blocks, calculated the standard suite of local field potential features, so band-limited power, coherent, all the usual sort of things. The other key thing is they do a clinical interview while they're there, and that is videotaped. Okay? So a key piece of data. You can see here the computational modeling on the bottom from just the sagittal view in this patient. You can see we're getting good engagement of cingulum, uncinate, forceps minor, and the frontostriatal fibers. So we're seeing kind of all the things that we want to see to know we're hitting in the right place. At 24 weeks, 5 of the 6 people are what we're going to call typical responders, meaning they got better. 4 of those 6 are in remission, meaning their Hamilton is lower than 8. The 4 that we don't have good LFP data for were also typical responders. So overall in this cohort, 9 of 10 were typical responders. We have 1 relapsed responder, which is a very interesting case. She started, after surgery, she started in remission, went most of the way through the trial, and then had a combination of things happen, some personal things, and fell into a deep depressive episode right around the 5-month mark. She eventually was treated, and at the 7-month mark was also a responder, but at our 6-month endpoint, she was not. So what's really interesting here is we have a cohort where almost everyone got better, so we can study the differences in how they got better and the time course of that trajectory, and then we have 1 person that basically drove that road in reverse, started better, and then got sick. So we have this really interesting data set. If you look at the individual traces, you can see there's tremendous variability. Okay? Some people get better quickly, some people take much longer. We see in red there our relapsed responder. We see one interesting feature, which is after an initial decrease, the Hamilton score, which is what I'm showing you here, often has a bump up a few weeks later. We'll talk about that. Okay, so idiosyncratic trajectories. So what do we do with this? Should we be trying to find a biomarker for the Hamilton? The Hamilton has a number of known issues, bias issues, the nonspecific symptoms that we talked about. Essentially, it's capturing things like their local mood day to day in addition to their disease state. So normally when we look for a biomarker, we want the biophysical signal on one axis and their ground truth on the other axis, but it brings this question, what is our ground truth? If you look at the clinical notes, here's one patient. They seem to have an increase in their Hamilton score. Those red dots are where the clinician decided to increase the dosage. You see highlighted here is an area where their Hamilton went up and the clinician didn't increase their dose. If you look into the notes, they say, yeah, I'll paraphrase here. The clinician essentially says, yeah, their score's going up, but they're really just anxious. They're feeling their depression go away and they're not sure who they are without it, and they're worried that they're going to feel alone. This person, they're not getting worse in their depression. They have something else going on that's going to take therapy to recover. This person is the same sort of thing. Depression metrics spiked up, the Hamilton spiked up. If you look at the clinical notes, there's a very stressful personal experience for this patient that they describe in detail, and then the clinician says, essentially, yeah, this is anxiety. Like this person had a really bad week. They had some family drama going on. They're not sick, they just had a bad week, and it gets reflected in these measures. So what do we use for our ground truth measure? Well, we know that in the first month, all of these patients are sick. If you look at their aggregate Hamilton scores, there is no clinical disagreement that they are all sick in the first month. There is also no clinical disagreement that they're all well in the last month, in month 6. And I'm talking about the typical responders here. We'll take our relapsed responder out for a minute. So what we're going to do, we're not going to try to regress against the Hamilton. We're going to build a sick-well system. That's what we want our biomarker to key on. Is this person sick? Are they well? Where are they at in between those two states? So we're going to use that data. It's not clear whether we're going to see a change in the subcallosal cingulate, remember, because it's a network disorder, but we're going to build a neural network classifier to try and see if we can detect it. In particular, we're going to build one neural network classifier for all 6 patients. And what you see is we can actually get quite good discrimination performance. So there is something going on in that data that is a marker of whether that patient is sick or is well. Okay. Area under the curve here of 0.87. So quite good performance. Right? And again, a single classifier. So whatever is happening, there are features in common across all 5 of these patients. It doesn't tell us what those features are. We could try a probe. There are ways to probe and say, what's your most important feature? But it doesn't really give us a full picture of what's happening. So we need now to turn to trying to explain this classifier. So we open our machine learning textbook, right? And we say, well, how do we explain these things? And the standard answer you're going to get is black box. We don't know, right? So we're going to move to a notion of what does explainability mean? So we need to sit down and think about this. We're going to abstract our problem here to a classifier that's going to classify these fruit shapes for a minute, and then we'll get back to some brain data. But this will be easier to illustrate. If we're putting a bunch of images of fruit into a black box classifier that we don't know what that classifier is actually using to classify, we just get the class labels on the output, you might ask yourself, well, I don't know what it is. Is it color? Is it shape? Is it some other feature that I don't have a word for? I don't know. What does it even mean to explain this classifier? We're going to use the language of causal statistics. We would like to identify aspects of the data that have a large causal effect on the classifier. Meaning, if we intervene, if we were to change that aspect of the data, then we would change the classifier's output. Okay, so graphically what we mean here is if we take our data, we would like to organize it in a way that different aspects, in this case, classifier relevant aspects and non-classifier relevant aspects are on different axes. So you see here the divisions by color and by shape. And if we take one of these, say take the orange and say keep the shape, so be an apple, so roughly the same shape, but move the color from orange to red, and then look at what happens when we probe that classifier and see, oh, your label changed. You went from one class to another. Whereas if we would have changed from carrot to orange, we wouldn't have seen the same change. This is notionally what we want to do here. Okay? So how do we do that? Well, the tool that we've developed is called the Generative Causal Explainer. Essentially, we set up a causal statistical model where we have two sets of latent variables, alpha and beta. Have in mind your favorite dimensionality reduction approach. Think PCA, if that's easy for you. We're going to go into the principal component space, and we're going to divide the principal components into two subparts, alpha and beta. Those are going to combine together to form a piece of data, right? So we have two different aspects that are going to get combined together to form a data point, and then that data point is going to get put through a classifier. X is going to get put through to find a class label Y. Okay? We're not looking for an individual feature. We're looking for something like an axis in a dimensionality reduction algorithm, something that could be complicated. So this is actually how we set it up. We train another machine learning system to work cooperatively with the black box. So the classifier is trained, that is fixed. We are not changing that now. But we're going to train another machine learning system whose job is not to classify. Its job is to generate data. And it's just going to keep generating data and pumping it through the classifier. But we want to train that system. Okay, so again, think about calculating PCA. And we want to train that system so that alpha, the dimension that we select to be the classifier relevant one, actually has a causal impact on the output of that classifier. The other one, beta, is used just to help capture our variance in our data. Okay? So in this case, we're not going to use PCA. We're going to use something more nonlinear. It's called a variational autoencoder. So a very kind of nonlinear neural network-based method. But essentially, you can conceptualize it like that. Dimensionality reduction, where some of our features have been trained in order to have a causal impact on the output of the classifier, and the others are left alone so they can just capture data variance. I'm not going to belabor this because we started a little bit late, but I'll say that we evaluated this in the kind of standard way that we evaluate machine learning methods like this. So we put data in, we trained it up on a classifier for digits. If we move the relevant variables, we get things changing their character, like from an 8 to a 3. If we move the non-relevant variables, we get line thickness changing and slant changing. We can do the same thing with different data sets, and we can do ablation studies where we take those variables out and make sure that it's actually what the classifier is depending on. So for now, we're just going to say that we evaluated this as a whole separate paper. So let's go back to our data from these patients. We've already shown you the trained classifier. Now what we're going to do is we're going to apply this to the classifier that we learned on the local field potential data from these patients. Then we're going to look at what those discriminative components do during the intermediate weeks. So the weeks when we don't know whether a patient is sick or well. So SDC is the acronym here, spectral discriminative component. So that is the dimension in this generator, right? That is the explainable variable. And we call it spectral because the features that we're using are largely spectral features here. Okay. What we see, I'm showing you all 6 patients here on the right. In green is their SDC. So this is the LFP-based marker. And in orange is their Hamilton score. So you can see, first of all, roughly, they're moving in the same direction. They seem to have good kind of qualitative correspondence. There are some really interesting individuals to point out. Here's our relapsed responder. You can see the Hamilton crashes around week 15 or so. So notice that we could have identified that crash coming approximately a month to maybe 6 weeks before they actually showed symptoms that were picked up clinically. Something was happening with this patient in their brain. And this marker, even though, let me remind you, this marker was not trained on this patient at all. All of the training for the data pipeline that I've shown you was done on the other 5 patients. So this is a complete generalization from the training that we did to a patient that our algorithms have never seen before. Also, remember our patient that was having anxiety because their depression felt like it was lifting and they were uncertain how they went forward. We can actually dig into the Hamilton subscores and we can see that that's what's happening. Notice that their brain is still moving in the right direction. They're still headed down, which in this case is going from sick to well, just like the Hamilton. And we can see before this point, their depressed mood was a much bigger factor in their overall Hamilton score than their anxiety. After this point, their anxiety is on par with their depression. So something is happening with them, but it's not a worsening of their depressed mood, and the brain reflects that correctly, that there's something else going on. If we look in the aggregate, we can see across patients, we have a signal that has lower variability than the Hamilton. We see an interesting point around week 6 or so where there's an early transition where they dip below 0.5. They kind of dip below threshold and look a little bit more well, and then they come back to being sick for a while until they eventually decrease into stable wellness. Note that if we try to correlate this on a person-by-person basis with their Hamilton scores, it does not correlate. We are not tracking the individual fluctuations in the Hamilton. If we look instead at a notion of stability, so take our stability criteria. Remember, in the Hamilton, if we drop by 50%, it's considered a response. So we're going to define stable wellness as where do they get below 50% and stay there for at least 3 weeks. So they've gotten into the response range and then they stay there. We're going to do the same thing on our LFP marker. And so you see here on the right, the individual sick transition to well for all of our typical responders. Okay. You can see there are a few patients that are off by a little bit. I can talk about them offline, but overall accuracy is almost 80% in identifying week to week whether they are sick or whether they are in stable wellness. So here we have our first clue that we asked for in our biomarker, right? We are corresponding to something that is behaviorally relevant. We are corresponding to the transition from sickness to stable wellness. Let's take a look at actually what's going on, because I promised you we're going to explain something, right? So we can actually look at these features. We can put a signal in, get into our latent space, and then we can move. We can go to one principal component effectively, and then we can move along that axis and say, what happens? What are the features that seem to be, the classifier seems to be keying on? So here you see a plot of all those features, and the ones that are statistically significant are in the left hemisphere, alpha and low and high beta, and on the right hemisphere, high beta and gamma. Those are the features that over the 6 months of therapy seem to be what the classifier is keying on. If we look just on the left side, for instance, we can see that the 5 typical responders all see substantial increases in their left, low, and high beta. Patient 1 there in the yellow is our relapsed responder. So she is supposed to be going the wrong way, right? Because that's actually what happened with her clinically. I want to point out, first of all, we weren't sure we would see anything here, but second of all, this is certainly not what was expected. From interoperative data, so when you're acutely recording, you implant the electrode, you provide therapeutic levels of stimulation in the operating room, and people were able to measure and see actually what you see is a decrease in beta. So the acute effect of stimulation when you first apply it is a decrease. If we look at our 1-month data, we see that as well when they get turned on chronically. And in fact, that decrease in beta corresponds to how much their Hamilton has decreased 1 week later. That's what this graphic from Sendi et al. Is showing you here. If you didn't know anything other than the acute response in the operating room, you would have expected that we should have been chasing a decrease in beta. Okay, if you were building a closed loop system, you would have been going the wrong way. You would have been chasing the wrong direction. The signal that we need to get people stable well is the opposite direction from what happens with effective acute stimulation. It's long been suspected that the recovery of depression patients to DBS is due to some type of adaptivity, some type of plasticity, and this is the first concrete evidence that we have of it. That something different is happening over 6 months than happens over, the minute timescales of acute stimulation. We can actually look and see what happens when there's a therapeutic intervention. We have time points where the clinician decided to increase the dose on the patients because they weren't doing well clinically. What I'm showing you is all of those points of stimulus change aligned to the week 0 time point. So week 1 is 1 week after stimulation change. Week -1 is 1 week before stimulation was eventually changed. What you see is their Hamilton score does not causally move in the next week or two after stimulation has changed. So according to that gold standard metric, they actually have no causal effect in the week or so after a stimulation change. Whereas there is a statistically significant improvement in the LFP signal in 1 week after stimulation change. So what we see is the second thing we needed, which is movement with a therapeutic intervention. So we have at least our two basic ingredients to try and have a biomarker that's actually useful to tell us objectively what's going on. I want to make just a couple of connection points. I told you we also have the face data sets. We have videos, essentially, of these patients during their clinical interviews. And we all saw it. You can see when they're sick or when they're well. We don't need fancy tests for it. We did essentially the same pipeline, but using machine learning classifiers on their face. And we can identify features. This is a patient in the beginning and in the end, first and last month. You can see his face looks different. But we can also identify which action units, which muscle groups seem to be changing the most as he gets better. We actually have quite good classification just based on the face. So based on the face, we can tell whether an individual is sick or is well. I do want to make one note here. There's much more idiosyncratic character to the face. So this is an individual classifier built for each person. So it's not the same as the brain signal where we were able to build one classifier that worked for everyone. But notice we have a lot of things happening. Here we've colored green things that are in the upper face, purple things that are in the lower face. So notice it is not that they are just smiling more, right? It's a complicated change in their face. We see here in the aggregate, I won't belabor this, but if we look at that face classifier, next to our brain signal, they seem to be moving in concordance, even including our relapsed responder. If we look at the time point where their face becomes stable and their brain becomes stable, we see concordance. We get a Kendall's tau of almost 0.9. So rank order, the people that look better first also have a brain signal that gets better first. We can also look at their pre-surgical DTI imaging. We can look at the health of their white matter tracts. So measure that with fractional anisotropy, FA. It's a measure of white matter health. You can see here, if we look just within our networks, you can see, again, forceps minor, I'll try and point out. The blue areas are areas where there is significant correlation with the time to achieve stable wellness in the patient. So big piece of damage here in the left forceps minor, almost out to the frontal pole. We have a big piece of damage here in the posterior cingulate cortex. Another one here down by hippocampal formations. So essentially, what we can see are areas, and it's what's shown on the plot in the bottom, where the more damage you have in those areas, the longer it takes you to get better. We don't know exactly that it's a remyelination process. That's certainly one possibility on the table. But the more damage there is, the longer it takes you to get better under stimulation. So what we see is this is the first longitudinal study of chronic stim in an SCC DBS patient. We didn't know what we would find. Our explainable AI techniques have shown us a putative biomarker, which is it's a critical step toward scaling this therapy and moving it toward potentially another clinical trial. There's evidence that we have multiple things going on. We see acute relief, then increase in some types of symptoms, but they seem to be core anxiety symptoms and other things rather than core depression in everyone, and then a longer-term recovery. There's a lot to learn about this. There are potentially multiple mechanisms going on. This would actually make a lot of sense given what we know about the pharmacologic agents. We have fast-acting agents like ketamine that last 2 to 3 weeks and then have to be readministered. We have slow-acting agents like SSRIs that take 4 to 6 weeks to reach their full effect. They have different types of responses for the patient. They also act on different mechanisms. There are also some interesting correlations with the specific frequency bands that we're seeing, the beta range and the gamma range. Certainly it's been hypothesized that there are connections between those ranges and the things that happen in depression. So broadly, if you're familiar with that literature, you might look at this and say that this is evidence supporting that an initial response is due to a disinhibition globally, and then a slow restructuring of inhibitory control over the time period of recovery. Again, this is really just kind of hints. We don't know anything about this, but if you're an expert in those areas, just giving you some hints in case you want to talk about it later. We've seen here a really interesting connection between the electrophysiology, the behavior, and the anatomy, right? All these pieces are coming together. There's more to explore, but I just want to remind you for a second that we saw in the face that what was mediating the change in their face to look more well were mostly upper facial features. And in the imaging, we saw, among other things, deficit in the posterior cingulate cortex, especially more so than anterior cingulate cortex. Just a hint at something that we haven't explored fully. There are different control networks for the upper and lower face. This has been shown in human, it's been shown in non-human primate. The upper, called the primary emotional system, this is kind of the real emotion that you're feeling. Lower is the social emotional system. This is you're forcing yourself to smile. It's the fake emotion. This is why people tell you to smile with your eyes, right, when you're getting a picture taken. Different networks that mediate it. I will just point out that the networks that mediate the primary emotional system, and here on the right is shown in non-human primate, but it's very, very similar, are higher motor areas, okay? So supplementary motor area, M3, and so on. In human, M2, M3, M4, okay? These are, either adjacent to or part of cingulum bundle as you head toward the posterior cingulum tract. Okay? The social emotional network is controlled more by premotor cortex and M1. Both can have some limbic involvement, but these are further away from our stimulation network, right? So we certainly weren't expecting this, but in hindsight, it certainly makes some kind of notional sense that we might see more recovery in upper facial features than lower facial features. So to kind of step back for a second, we've talked a lot in most of our time here together about peeking inside the artificial intelligence black box. But I do want to say there is still this problem, right, of how do we actually measure behavior from these people? We have these kind of imperfect scales, these Hamilton scales, and again, they're trying to describe how they're feeling and they don't have the language for it. So how do we deal with situations where there's a limited language to describe the internal thought, the internal percept, whether it's a human or an animal? When we have to deal with things like this in the machine learning literature, and by things like this, I mean we want to characterize data, but we don't have a good feature space for it, and we may not even be able to learn a good feature space for it. What we do is we look at techniques like similarity embedding. We say, listen, I don't know what the right features are, but what I can do is maybe I can lay this data out such that things that are similar are close together and things that are dissimilar are far apart. And similar and dissimilar are completely subjective judgments, so that's being given to you by a human, right? So for instance, if a machine were to lay out all these images, they'd put things together maybe like this. You see a lot of color coordination. But if you asked a person to organize things by gustatory similarity, what tastes would want wasabi and guacamole very far apart, and the computer would have put it very close together. We have no way to get at that relationship other than querying the person. The problem is it's very difficult to query people for things like this. This actually is very similar to a notion that's used in neuroscience really a lot in the last 10 years, which is representational similarity analysis. This is used when you have electrophysiology data and a computational model like a deep neural network. You want to say, listen, the feature spaces are probably constructed very differently, but we want to see if there's a similar organizing principle. We can't just put them side by side, but let's see if two things that are similar, two stimuli that are similar in the electrophysiology are also similar in the model. And that's the way that we're going to try and correspond these representations together. It's very similar to that, only it doesn't get used with behavior very much, again, because it's difficult and time-consuming to collect this data. We actually sort of know what to do, right? This is a psychometric sort of task. The classic task would be what's called a triplet query. You pull 3 data points out of your data set. You make one the reference, right? And you say, here are 2 possibilities. Which one is closer? It's more similar to the reference, right? And you have them pick between the 2, so they have a binary choice. This works. If you collect enough of this data, then you have essentially a dissimilarity matrix, and you do dimensionality reduction, and you can get this type of similarity embedding. You can make a mental map of the space of the person's subjective percept. The problem is it takes forever for any reasonably sized data set. It's entirely impractical. I'm not going to dive into the details of this, but I'll just say we have new state-of-the-art methods that we've developed for doing this better. One key thing is that they're active learning methods. So what we do is we look at the answers that the subject has given us before, and we pick new queries using information theoretic criteria to say, what's the most informative query we could ask right now? So it lets us ask many fewer queries. We also extended the query type to be these rank ordered lists. So instead of just a binary choice, we can give them a list and say, will you rank order these with reference to similarity to this reference or this anchor object? And we show people can do that out to lists of 7, 8, 9 objects, and doesn't seem to be a problem for them at all. And we get much better data that way. It makes the people be more self-consistent when they're answering. And so it's not just that you're more efficient in collecting the data, it's actually higher quality data. We've done this, we collected a data set of almost 700,000 queries, or I'm sorry, 200,000 queries from 700 subjects. This is just to show, like, our methods are much better than the randomized query selection that had been done before. So it opens the door to doing this practically. We have a tool. Really notice the AI system doesn't touch the data at all. We don't need the AI system to know anything about the images, about the taste of the images. The only data that we work with is the response from the individual. So this could be anything. This could be your favorite sensory stimulus. This could be a motor situation that you want people to compare. Hey, was that movement similar to that movement, even though your joint angles were all very different? It could be an interoceptive sensation. If that feeling you're having right now, is that more like yesterday or more like the day before that? Really, the algorithm doesn't care what sort of data it is. It's just going to figure out which points in that data set to ask to most efficiently make a mental map of the space for the subject. Easiest with humans, not impossible that you might be able to do this in some animal preps. It doesn't necessarily have to look exactly like this. We have some modifications to it that we've talked about actually possibly doing with animals. You wouldn't have them rank order lists, but maybe still picking most similar sensory stimuli could be something that they could do. I just want to show this to show how abstract pilot set that we just collected, we've barely analyzed. This is asking people to take day-to-day situations and choose how similar they are in terms of their perceived effort. Remember our core symptoms of anhedonia, lack of interest in doing things, psychomotor slowness, this huge perceived effort. We're trying to get at whether we could characterize depressive symptoms with methods like this. This is just a basic clustering applied to it. I just really wanted to show you that we could go to very, very abstract queries like this and people can do it. We don't know yet what it means. There are a few kind of idiosyncrasies in the clustering that we don't know if they're real results or not, but happy to talk about that offline. I think what I'm trying to show you here is a way to try and use AI techniques to give voice to people who don't have the language to describe their symptoms. Remember, I said that that's also what good art can do. So as we try to put these pieces together, I think we need to remember that not all of our solutions are technical, that art still has a role. So just a brief advertisement here that I've been working for the last year and a half on a newly commissioned piece of modern dance with GT Arts and a local modern ballet theater called Terminus and a choreographer named Troy Schumacher from the New York City Ballet. Making a piece of modern dance that's focused around neuroscience, neurotechnology, and neuroethics. Trying to give voice to those things that we don't yet have language for. Some of you have met with them. It's basically an artist in residence sort of process that they've been going through. We'll have the premiere here in the fall. Be sure to advertise that broadly. It'll be here on Georgia Tech's campus, and I'm really excited about it. Just want you to hear from some patients what it sounds like when they come out.
Incremental changes. Things got a little bit easier. And even in the smallest things, it got a little bit easier to brush your teeth. It got a little bit easier to get out of bed. It got a little bit easier to have hope. It just started a cascade of positive instead of the cascade of negative. And that was the difference. DBS didn't make me happy. DBS, it didn't solve all of my problems with how I was thinking or how I was behaving or my lack of boundaries and relationships, is it didn't solve any of those things, that needed to be worked on. All it did was make it possible for me to not have this huge burden preventing me from being able to solve all of it.
I walk more than I did before just because places I've been, I don't even think twice about walking to Emery Village anymore. Over here, it's just kind of like, here we go. But I'm not worried that I'm going to slip back into some kind of awful mood. I feel that I am better equipped to deal with the everyday things that happen in life.
So hopefully you see it in their faces. You hear them describing movement. You hear, especially the second patient, describing stability. Right? I'm not worried anymore. Styron's words, it can close us out here. For those who have dwelt in depression's dark wood and known its inexplicable agony, their return from the abyss is not unlike the ascent of the poet, trudging upward and upward out of hell's black depths. The poet here, of course, is Dante Alighieri. He's describing the Inferno. He quotes from the Inferno, and so we came forth once again, beheld the stars, and this is Doré's illustration of that moment there. So as we search for our Virgil, I hope we can use some of these technical advances as well as our connection to the arts and humanities to try and help these patients find a way out of it. Thank you for your time and attention. I know we had some technical trouble. I appreciate you sticking with. Thank you.
All right, so if you have questions, we can take them. You can just unmute and ask. You can put them on the chat.
I can't see the chat at all here, so just go ahead.
Yeah, Adobe, you can monitor.
Yeah, we'll monitor the chat here.
I can ask a question online first. Hey, Chris, thanks.
And maybe, Chris, I'll ask for people that are here, you can repeat the question so that people can hear up there. I was wondering, I hear sometimes in these, at least these trials with depressed patients who have TMS stimulation, that there's significant placebo effect, effect in the placebo group, something like 25%. And I was curious whether or not this sick well metric that you have trained on the actual patients that receive stimulation might be able to be applied to patients that get better in the placebo trial.
Maybe, and we can certainly look. You're talking about in a DBS placebo group, because we couldn't get our data for this in a TMS trial because we need the invasive recording. Yeah.
Curious, or whether it might reveal some kind of problem with the Hamilton scale, even if you could.
Maybe. Yeah, maybe. Certainly, there are a lot of things we could do there, and I should say we are looking at EEG correlates that wouldn't necessarily need the invasive recording, but we're not there yet. But we're looking at those sort of things. Maybe it'd be very interesting to kind of look at all the subscales in the Hamilton, see if we're seeing factors in the brain signal that correspond to different sub-factors in the Hamilton score. We've looked at that a little bit. I can't say that anything groundbreaking has come from it, but yeah, the placebo effects are real. These patients, they desperately want to be better. They they want this to work. This is a treatment of last resort. They're working closely with a clinical team, and they want to, they're willing subjects. They want to please their clinical team. So there are all sorts of reasons that you can get placebo effects here too. Be very interesting to know. Right now we have no placebo patients, right? We have no shams in this group. It'd be very interesting to know if we had that data, whether when we see potentially placebo-induced improvements in the sham group, if their brain is also moving in the same direction that the treatment group is moving. But we don't have that data. It would take another clinical trial to get that. Yeah.
Simon, go for it. Just speak as loudly as you
I
can.
forgot to repeat the question. I'll do better at that. I'm sorry, people online. Yeah.
Actually, we could hear the question pretty clearly online.
Do we have one in the room here?
Do you want me to ask?
Hey, Simon, is that something? Yeah, go ahead and start. I can hear you now.
Okay, great. Hey, Chris, thanks. That was really fun. I really enjoyed the talk, although it had some some sobering messaging as well, but it was really, really interesting. I was thinking a little broadly when you were talking about this stuff that depression is one of these diseases that you were talking about that is diagnostic, meaning that we could have a lot of different physiological origins and signatures and causes and comorbidities and such. And you know, as you know, a lot of diseases are like that, migraines, things like that. And so for these kinds of diseases and problems, do you think that you could discover with some of these methods structure? Yeah, obviously not with the small group that you did this intervention that you were talking about with, but if we got this to much larger populations, could we use some of these techniques to sort of discover patterns and features, latent diseases within those sort of broader diagnostic categories? And is anyone thinking about doing that?
Yeah.
That space?
I think it's a really, really great question, Simon. So, you know, I mean, one thing, even if you just narrow your question back to depression for a second, you know, there's a core question there, which is every patient different? Or are there phenotypes, right, that we can group people into that would respond to similar treatment and they have similar symptom clustering? In some ways, the last work that I showed is one attempt to sort of say, maybe we can talk to them behaviorally and find some symptom clustering rather than just using one-dimensional, numerical scales, and understand what the phenotypes are. I think the data, maybe we could look at that. We certainly haven't, and as you point out, it's probably too small of a group to do that. I will say the next generation of devices from Medtronic has more steerability in the current. And so it will open up an adjacent question, which is, you know, we don't know that you need actually all four of those bundles. And it's possible that different phenotypes of depression or different comorbidities with the depression would correlate to different parts of the network. And so in a larger trial with more steerability, you know, it's something that you maybe could dissect out a little bit. And we're certainly not there yet, but I think that is a super, super interesting question. Definitely on my radar for next steps. Thanks. One last question here in the audience. Yep.
Yeah, my question is in relation to, you mentioned early on the statistic where based on socioeconomic status, depression overwhelmingly affects those in poverty. And I was curious to know kind of what the demographics are for this particular population. And yeah, I guess if that is homogeneous, what sort of efforts can be made to make sure we include those in poverty in these sorts of trials to try and understand the differences that we'd observe?
Yeah, in a large-scale clinical trial, I'm certain that, I believe I am unmuted, yep. In a large-scale clinical trial, I would hope and imagine that would be part of what they try to achieve in getting a representative sample. You know, in this population of 6, I actually don't know what the socioeconomic variability is. I know of at least a couple of the patients in the next cohort that I didn't report on that are in very challenging economic situations. Of course, you know, by the time they show up at our door, many of them, you know, have some affluence because they've been seeing doctors and doctors upon doctors. They've gotten to the point that they're treatment resistant. They haven't abandoned the notion of therapy. I mean, for most patients, they have some level of resource in order to navigate through that. I can give you a chart of the demographics. Otherwise, you know, there's a pretty broad spectrum of age range. I believe the 6 that I showed you were 4 female, 2 male, which is, you know, broadly reflects the gender, the gender bias that you see in depression diagnoses anyway. But I don't have socioeconomic data on them. I would guess that it is more homogeneous than what one might like in a large-scale clinical trial. But good question, certainly important.